Well, thank you, and thank you for the invitation to speak. It's always fun to bring this to this particular audience. So today I'm going to talk about something that's a little different than I normally talk about. In fact, it was a side project that's been going on in my lab since I moved to Colorado State. And I use it as an example for why people who serve on graduate student committees should pay attention and talk to the students that they're co-advising, because you might step into a project that's really interesting and exciting. I was a little ambitious when I sent my title to Susanna. I'm not going to touch on the environmental influences because of lack of time. So today I'll just talk about the multitrophic interactions. But I want to give you a few definitions of where we're coming from, because the term phytobiome, I know, isn't a term that slips off your tongue. The phytobiome we see as the system of agriculture. It is the interaction of all living organisms that impact or are impacted by plants. So this includes, as a subset, the microbiome. So the plant microbiome is a component of the phytobiome, which includes plants, insects, nematodes, worms, soil, and the environment. So the thing that we are trying to engage people in is embracing the complexity of this system. I'm a plant pathologist by training, and historically we think about one pathogen in one plant, or maybe we'll think about the pathogen and the soil and the plant. But rarely do we consider all of the interacting partners. And many of you are familiar with the old Indian tale of the 7 blind men who were trying to say what an elephant looked like. And so I adapted that to the phytobiome. And so we look at this interaction and we say, well, maybe it's a bird if we look at just the birds that are flying around, or maybe it's a fungus, or maybe it's bacterial that is the reason for the interaction response or the phenotype that we're seeing, or maybe it's nematodes, or maybe it's soil, or maybe it's even a virus. But really, we need to think about this as the whole system, because in the environment, a plant doesn't just interact with one bacteria or one fungus or one insect. So with that complexity in mind, I want you to think about even deeper who's in that phytobiome. So again, we often think about, and I think that Dr. Seuss was brilliant, because in Horton Hears a Who, maybe many of you don't pay attention to Dr. Seuss, but I don't even have kids and I love Dr. Seuss. My husband makes fun of me. But Horton Hears a Who. Horton heard something on the pollen that was sitting on a flower. And it was a whole community of organisms, very much like what we think of as the microbiome or even the phytobiome. But think about it, even within that community, in that community of Whoville, the organisms, for example, the nematodes in the soil, they have their own complex of organisms that are associated with them. So it gets more and more complicated. So not only do we need to think about, for example, the microbiome of the soil, we need to think about the microbiome of the nematodes and the worms and the amoeba in the soil. And those organisms, in fact, do a great job of grazing on the bacteria, and how are they impacting that community? Same thing happens on leaves. So it's much more complex, and we need to embrace that complexity. So today I'm going to talk about a tripartite interaction. I'm still being a reductionist. I'm simplifying it to just a plant, an insect, and the community of organisms that live in that insect. But that's what my brain is capable of doing at this point in time. So we're going to talk about organisms that help insects in their need to colonize or to infest plants. And there's been some really wonderful work out of Gary Felton's lab that's been published that many of you are probably seeing, because they had a very nice paper in Science Reports just recently, where bacteria associated with the Colorado beetle larva manipulate plant defenses. So as the larvae are feeding on the plant, they're chewing and they're disrupting the structure of the plant. They're delivering bacteria into that wound site, and those bacteria induce a plant defense response, a salicylic acid-based defense response. And salicylic acid defense responses are antagonistic to jasmonate defense responses, which are inhibitory to insects. So in other words, the insect uses the bacteria in order to make the plant more accessible. They also, in this recent work, showed that different insects acquire different microbiomes based on the host plants that they feed on. So again, in this very important interaction, if you had only looked at the insect and the plant and not paid attention to its microbial community, you would have missed a very big part of the story. So I'm going to talk today about aphids. And aphids are very different than these chewing organisms. So aphids, and the aphid I'm going to talk about is the Russian wheat aphid, but I want to show you a video that'll tell you a little bit about how aphids feed on plants. So the aphid marches along the surface of the leaf, and then it inserts its stylet into the epidermal cells and tastes it to make sure it's on a compatible host or the right host. And when an aphid stylet penetrates the epidermal cell, for those of us who are not entomologists, it spits into the cell, and that spit sort of delivers, in some cases, aphid effectors, and then the aphid will suck back and test if it's the right environment. And so if it's the right host, then it continues to penetrate, and it moves between the mesophyll cells, the stylet. It's very flexible. It penetrates cells as it goes. And in fact, as it's penetrating those cells, it's inducing a very mild jasmonic acid response. It's considered that aphids are much more like, and then once it reaches the phloem, it penetrates, it spits into the phloem, and it decides this is where it wants to stay, and it continues to feed. And it continues to spit and then suck. Now, many aphids will deliver effectors, which are the proteins that sort of cause the symptomology in the plants. Interestingly, the Russian wheat aphid, which is going to be the system that we're going to talk about today, does not deliver such effectors. We know that from our work and from the work of others. So when I first went to Colorado State, I was invited to serve on the committee of a young man named Leon Van Eck. And Leon was interested in how aphids cause symptoms on wheat plants. And this is the Russian wheat aphid, which is an invasive species. And in fact, for those of you from Australia, was just found there recently and promises to be very devastating there. But so Leon did what many folks at that time were doing, which is he collected saliva. So you construct these little dishes with parafilm on them. You put the aphid on the top. You have a sucrose diet underneath. The aphid spits into that sucrose diet. You collect the sucrose diet and do a proteome analysis. And what Leon found is that he didn't detect a single aphid effector in that diet. Not a single aphid effector was spit into this diet. And this actually has been found by others more recently. But what he did find, so the only aphid proteins are this very tiny 1% of the proteins that he found in that diet. What he did find is a lot of bacterial proteins. Now, initially, people were saying, oh, this is contamination, but that's why me being on the committee as a microbiologist, sorry, that was for emphasis. I'm a microbiologist by training, and I'm sitting there thinking, wow, look at all these bacterial proteins in here. And interestingly, these bacteria fall into particular groups, which most of them are Enterobacteriaceae, which are often plant-associated bacteria, and in fact, often plant pathogenic bacteria, which is where my heart lies. So 99% of the proteins that aphids spit into the cells are bacterial proteins. So that led us to the question of, well, do the aphid stylets harbor bacteria? Now, as Leon said in his defense, bacteria or aphids don't brush their teeth. So yes, indeed, there are a lot of bacteria associated with the aphid. So this is the aphid here. You can see its stylet now blown up over here. And then as we blow up further, you can see that there are bacteria sitting on the surface of the stylet. So we don't know if the aphid is delivering the bacteria by spitting or if it's carrying the bacteria on the surface of its stylet, one of the questions that we want to ask as it's feeding. So then Tony Campillo came into the lab a couple of years after Leon left. We sort of sat and pondered about this for a while. And we knew that these bacteria were probably responsible for aphid feeding, but we didn't quite know how at the time. So Tony was a microbiologist, and he came into the lab and working with Emily Luna, who you'll hear more about later, decided, okay, let's see if these aphids have bacteria associated with them. So he fed aphids on the artificial diets and then looked in the artificial diets for living bacteria. He crushed aphids and looked for bacteria, culturing bacteria, and he looked at leaves that had been fed upon by the aphids for bacteria. And sure enough, we were able to identify a whole group of bacteria by culturing. And in fact, most of these were Enterobacteriaceae, a few were Excellaceae. And what's most interesting to us is that what we found by isolation recapitulated what we found in the proteome. So we found a lot of Erwinia, Pantoi, Acinetobacter, Enterobacter, and a few others. Now these down here are often symbionts. They live in the aphid gut. And this is the tricky thing about aphids, is they have a group of bacteria that live in their gut that are essential for them to survive, that they make amino acids that the aphid needs. So when you're treating aphids to get rid of the bacteria, you have to be very careful about those. So I'm sorry, I meant to show you the different groups that we found. So then Emily Luna took over the project, and she asked the really important question, and that is, do the bacteria that are associated with the Russian wheat aphid enhance virulence to wheat? And fortunately, Emily is an entomologist by training, but has some bacterial experience working with Tony. So she was able to do what we needed to do, which was clean the aphids up. So the first thing we did was treat the aphids with antibiotics. Now you have to be really careful, as I said, not to kill their internal populations of bacteria. And we were able to do that. And we showed that the aphids did cause less symptoms. But we were concerned about the criticism of you're affecting their Berknaria and the other internal populations. So working with Emily, we decided we would try an experiment to see if the aphids picked up the bacteria by feeding next to their mothers after they're born. Now, aphids give live birth, which many of you may not realize. And so what Emily did is she sat with a brush and picked out babies, abducted them, kidnapped them as soon as they were born, and established colonies from those aphids. And what do you know? They were clean, pretty clean, relative to the ones that were allowed to feed next to mom. So we established colonies, which you can keep clean for quite a while. I was, as a microbiologist, shocked at that, for at least three weeks or so. And then she infested plants. And what this shows you here is that the clean aphids did indeed have less bacteria associated with them than the dirty aphids. The dirty aphids being the ones that fed next to mom. So we just recently did a microbiome analysis of the aphid. And I wanted to show you, we compared the dirty and the clean aphids. And what you can see is that, I won't show you all the nitty-gritty, we're still analyzing the data, but the dirty aphids are, well, first of all, I should tell you that Berknaria, which is that endosymbiont that's crucial for the aphid to survive, dominates in both. But if you look at the other bacteria that are associated, the dirty aphids have more of these Enterobacteriaceae that we're interested in than the clean aphids. And they also have more of the Moraxellaceae. So the very ones that we were seeing associated with aphids from our culturing techniques are the ones that we're finding. Also, the diversity of the population, as you might expect, on the dirty aphids is much higher. So the bottom line is that the clean aphids, the ones that were abducted from mom and used to develop the colonies, cause much fewer symptoms on wheat than do the dirty aphids. And the question that an entomologist would ask is, well, was the aphid still healthy? And yes, the answer is we got the same number of aphids off the plants after they had colonized and infested the plants as we did before. So how do these aphids, or these bacteria, help the aphids to colonize or infest the plants? So we went in and mined the data from an old transcriptome analysis where folks had taken samples of infested wheat leaves infested with Russian wheat aphid at 5 and 48 hours after infestation. And what we found was, in looking at the two biosynthetic pathways that are responsible for production of jasmonic acid, which jasmonate is the pathway that's induced by these feeding insects that damage and feed the leaves, and is a defense response pathway, and the salicylic acid pathway, which is induced usually by plant pathogens, and it's the more subtle pathway. And it's induced once the aphid usually penetrates into the phloem, is that both pathways, so the way this graph is the top half of the triangle is the 5-hour feeding, and the bottom half is the 48-hour feeding. So if you look at here, you can see that by 5 hours, both jasmonic acid and salicylic acid pathways were upregulated in these interactions. And by 48 hours, by and large, most of the jasmonic acid pathways genes were downregulated, whereas the salicylic acid pathway genes remained upregulated. Now, this is consistent with what others have found in other systems, that jasmonic acid pathways go up early and then down, and salicylic acid pathways remain high. Now, the salicylic acid pathways are those which are probably being induced not by the aphid, although the dogma is still out there that the aphid's inducing them, but possibly by the bacteria. But what these findings are consistent with is that the salicylic acid pathway might be antagonistic to the jasmonic acid pathway. So then we asked, are the hormones produced in these interactions? And what we found is that, yes, indeed, the clean aphids, which are the green bars, induce less salicylic acid than the dirty aphids, consistent with the salicylic acid being induced by the bacteria. And the trend for jasmonic acid was not as clear. The trend is that they're lower at the later time points. However, notice that the levels of jasmonate that we were detecting in these leaves were very, very low, almost below the levels of detectability. So we can't consistently say that we saw a reduction in jasmonic acid production with the enhanced production of salicylic acid. But we do know that these bacteria are contributing to the formation of salicylic acid, which is antagonistic to jasmonic acid production. So our conclusion is that bacteria associated with the Russian wheat aphid manipulate plant defenses to enhance aphid virulence. And this is the hypothesis that we're moving forward on at this point in time. So by looking at this entire system of the phytobiome, we've been able to identify some possible targets for resistance breeding and biocontrol in the future. So if we had only, currently the practice is to breed for resistance to the aphid. Now, we don't know if the few genes that are out there to control Russian wheat aphid affect the aphid or the microbial population that's associated with the aphid. But what what I tell the breeders is maybe we're targeting the wrong organism. Maybe we should be looking at breeding for resistance to the bacteria so that it doesn't trigger the particular bacteria that the aphid is delivering, so that it doesn't trigger the salicylic acid response, which in fact will turn down the jasmonate response and enable the aphid to feed. So maybe we're looking at resistance. We could look at resistance in a different way, and in fact it'd be a lot easier to screen for these two bacteria. The other thing that we need to think about is, could we alter the leaf microbiome in order to, so that when that baby feeds next to its mom, it's not picking up these other organisms. Maybe we could outcompete the organisms that mom is contaminating the leaf with and prevent the baby aphids from becoming infested with these particular symbionts that are important. And third, possibly in the future, what we're looking at is, have these aphids actually evolved to rely on bacterial effectors? Now, remember at the beginning, I told you in the proteome we didn't find any aphid effectors, which is different than peach aphid or the other aphids that are out there. Russian wheat aphid is very interesting in that it doesn't deliver viruses. It's never been known to vector a virus, and it doesn't deliver effectors into plant cells. So maybe there's some association that has evolved where this aphid has become reliant on bacteria in order to cause symptomology in the plants or virulence. So finally, before I conclude, I'd like to advertise two things. One is a rice research to production course. You don't have to work on rice to come to this course, but you do have to be between 21 and 35. There are 10 fellowships available, and we've run this course now. This will be the 10th year for this course, and the students that I've had that have gone to the course say that it's a life-changing experience. It's 3 weeks in the Philippines at the International Rice Research Institute, where you do everything from learn how to plow with a water buffalo to do molecular breeding, all in 3 weeks. And finally, I'd like to thank my collaborators, my lab members. So Emily Luna took over this project and really has taken it to the next level. And then collaborators, Chris Argueso, Paul Odie, Anna Maria Botha, and Michelle Cilia for the proteomics, and Nora Lapitan. And the last thing is to advertise for you the Phytobiomes Alliance, which is an international consortium of folks that have come together, both industry, academia, and government groups, who've come together to try to pull resources from different agencies and governments towards understanding the phytobiome as a system. And you're welcome to contact Kelly Eversole if you have any questions. But thank you very much.
So what is the outcome of the aphid infection for the wheat plants? Is it usually, does it destroy the whole crop? Or is
it can, especially if it's a susceptible field. So that you'll see leaf curling and then streaking, and then they'll dry and they'll collapse. So it can be extremely devastating to a wheat field. In the US now, we have a few sources of resistance which have dropped the problem down to a manageable level. But in Australia, they're just beginning to see the tip of the iceberg, I think, because it takes a long time. You know, wheat is not like rice or a nice dicot. It's a difficult crop to breed. It's a hexaploid, and it takes time. I mean, that can be advantageous in certain things, but it takes time. So, yeah.
All right, we have,
Jan, are there any land races that are resistant of wheat that are resistant to the aphid?
Yes, there
what's
are.
the mechanism whereby
it's not known what the mechanism is. Even in the gene, there's a single gene that our breeders in Colorado have been using for some time, and they still don't know the mechanism for resistance of that. Again, I'd like to go into it more deeply, but wheat is such a complex crop. I'm trained as a microbiologist. I'm used to a haploid genome. I managed to work in rice a diploid genome, but a hexaploid, that's getting beyond my skill set. So, but it is that particular gene, we don't know the mechanism. And I don't know if it targets the bacteria or actually the insect. But we can begin to ask those questions now because we know how to make clean aphids, dirty aphids. We have the bacteria. We've actually named a bunch of new species of bacteria from the aphid, finding things that hadn't been found before. So we can begin to ask those questions.
Question over there.
Do you think that you could, now that you have your clean aphids, do you think that you could complement them by adding back in isolates from the dirty aphids to start to figure out exactly which strains are doing it?
And we've started to do that. We don't know whether it's a consortium that's important or that it's one or two. We know that the bacteria do induce salicylic acid, but we don't know if they work together. So one of the reasons that we did this microbiome study and looked at clean versus dirty is that we're seeing, we're hoping that'll help us target down which bacteria to look at, because not only were some of them more prevalent in the dirty than the clean, but some of them stayed the same level. So those that stay the same level probably aren't the ones we're interested in. So now we're using that as a filter to decide which ones we're going to go in and do the next steps. And in fact, those experiments will be starting very soon.
Hi, great talk. I might have missed it, but other than the salicylic acid induction or loss of induction, was there any overt pathogenic effects on the wheat plant from the clean aphids versus the dirty aphids?
They show minimal symptoms. They show a little bit of symptoms because they're not completely clean. When we put the bacteria that we've isolated, we've tried with several bacteria. We do not induce symptoms, interestingly. So one of the bacteria that we've characterized, Erwinia iniecta, because it was one of the most prevalent ones and we thought it might be an interesting one, does not induce disease on wheat at all, at high levels. But it does induce salicylic acid.
So did you say that you were not able to see an effect on the aphids from, comparing the clean aphids and the dirty aphids? The clean aphids were surviving and reproducing just as well as the dirty aphids. So.
Yeah, and that's
what are the
because we
implications?
didn't mess with their endosymbiotic populations,
Right,
presumably.
I guess that you're not showing the benefit of these aphids that, or the bacteria that is being introduced to the plants.
I see what you mean. Yeah, in terms of symptoms on wheat, we're not seeing any disease. I see what you're.
For the aphids for having
Yeah,
these bacteria.
so they're not able to cause symptomology on wheat. We see a minor reduction in the aphid population on the wheat, but we don't see, yeah, I see your question. Let me back up a little bit. So the aphids, we can measure their fecundity and their ability to survive on media, on the artificial media, and they're fine. On the wheat, they still build up some level of population because we haven't completely cleaned them up. Minor reduction, but they can still feed on the wheat. They don't cause symptoms. So apparently, we're not knocking back the aphid population, in other words, but we are knocking back their ability to cause disease or symptoms. Yeah, good point.
All right, we have one last question? All right, well then let's give Jan a hand.